Good morning, everyone. Good morning, Stavros, Shahab, Scott, Simon, and Phil. Welcome, everyone, and thank you for attending my master's defense on my research on the effects of moisture-induced thickness swelling on the microstructure of oriented strand board. I'd like to start by giving you a brief introduction on OSB. Most North American homes are built from wood and wood-based composites. And OSB production and consumption makes up for the largest market share, and it's expected to grow even further in the coming years. North America is by far the largest producer of OSB, with 85% of the world's production. OSB is mainly used for housing as a structural panel for sheathing, roof, subfloor, so during construction it can get wet in building sites. And that can cause swelling of OSB, which is a big problem because it can lead to unacceptable variation in thickness between and within boards, which may then need to be leveled by sanding, but that can be costly and time-consuming to do. Well, why does OSB swell? According to the literature, OSB swells due to the net result of the natural hygroscopic swelling of wood, which is reversible, the recovery of compressed or deformed wood, which is irreversible, the straightening of strands that might take place, and the lamination of adhesive bond lines. But are these all the factors? Well, we think there is something missing here. And if we look at another materials, such as clay soil, we can see how cracking can contribute to the increase of area in this type of material. As you can see, the blue portion around, which was representing the cracks. And volume change in silicic volcanic rocks is also associated with microcracks, as well as in sulfate-rich clay rocks, which has been shown to swell due to microcracking caused by wetting and drying cycles. And if you consider solid wood, there are some studies looking at checking or cracking propagation. And like in solid wood, when we look at a composite such as plywood exposed to wetting and drying, we can see that it also develops microchecking. These microchecks are different from lathe checks that is introduced during peeling when plywood is manufactured. And if you consider solid wood again, there are some studies looking at how modification treatments using microwaves can increase the volume of wood, in this case by creating numerous internal microchecks due to high internal pressure. So that leads us to our hypothesis, where we want to test if microchecking will occur and OSB exposed to wetting and drying. We also want to assess the extent to which microchecking contributes to the thickness swelling of OSB, and confirm the loss of zinc borate in OSB exposed to wetting. So the material we used to carry out this study is OSB used in I-beams, and it's made from a mix of lodgepole pine and aspen. The density of this material is around 700 kilograms per cubic meter. The adhesives used in this OSB are phenol formaldehyde and PMDI, and it contained zinc borate. So initially we obtained samples measuring approximately 50 by 50 millimeters and 10.5 millimeters of original thickness of the board. From those samples we extracted specimens measuring 20 millimeters long by 10.5 millimeters, which is the original thickness as I said, and a width of 2 millimeters. So a variety of methods are needed to understand checking of OSB. Firstly, to look at surface checking, we have developed a method using macrophotography that allows us to observe the mechanisms of swelling and shrinkage in real time during the entire process. So that gives us the full picture of the phenomena involved during wetting and drying. Then we used methods that allow us to look at internal checking. So we employed X-ray microcomputer tomography to look at 3D imaging of OSB. Lastly, we used field emission scanning electron microscopy, which gives us high-resolution imagery of the microstructure and enables analysis of the fracture leading to microchecking. So the first technique to look at surface checking, where we employed macrophotography. We have developed this device in-house. It is composed of a system that blows moist air during 2 hours to promote wetting, causing swelling, and dry air for 2 hours, causing shrinkage. The system allows high-resolution imaging of OSB through a macro lens that goes from 1 to 5 times magnification. The system has sufficient resolution to observe changes at the cellular level, but its field of view is large enough to visualize macroscopic changes in the structure of wood. And it can observe the swelling and shrinkage of OSB and solid wood specimens dynamically in real time. Then, the second technique using X-ray microcomputer tomography we used to look at internal checking of OSB. We used 2 X-ray computer tomography devices developed at the Australian National University to gather the datasets of the internal microstructure of OSB samples. So the first, a conventional system, and then the state-of-the-art high-resolution Helical X-ray CT. This device gives us better resolution and more signal-to-noise ratio compared to the conventional X-ray CT device. So we focused on the analysis of the datasets obtained by the Helical system. The time-consuming nature of the work and the resources devoted to look at just one OSB sample were enormous. It takes long use of instrument time for each scan and data processing, and the datasets are so big that it takes a supercomputer to reconstruct the tomograms, which then take months to segment and analyze them. So with the Helical X-ray CT device, we used the same sample before and after swelling, a cylindrical sample this time, measuring 10.5 millimeters by 10 as you see in the photo. The swelling was promoted by soaking the cylindrical sample for 48 hours under the water. And then we studied the microstructure of OSB using image analysis. So with the data sets from the sample before and after swelling, we selected the slice from 0 to 100% along the length. And then we analyzed the individual frames before and after swelling for each slice. Up to here, the phases that we can directly get using computer software or the wood, zinc, and airspace. But manual segmentation is required in order to threshold the voids and checks. We did that by using a drawing tablet with a computer. The third and last technique to locate fractures, we used field emission FE-SEM, and we had 3 treatments in this part of the study: control, water soaking during 72 hours, and the last treatment, which exposed OSB to the same cycle of wetting and drying as I mentioned before for the macrophotography. So after they were treated, I took small specimens from those samples and embedded them with a low viscosity epoxy resin under vacuum and pressure. Here is the process of embedding the specimens with the resin. And after curing them in an oven to harden the resin, we could sand and polish their surface without altering their original structures. We had 5 replicate samples from different panels for this technique. So now I'd like to show you the results of this study. I'd like to start by showing the results on the surface checking obtained by the macrophotography. So as you can see from this animation, which is a time-lapse from swelling and shrinkage, that it shows checking developing during wetting and partially closing during drying. Checks occurred at the interface between late wood and early wood and rays of softwood flakes, and less commonly in the rays of aspen flakes. We can see numerous checks in softwood here. And here we can see bark inclusion checking badly, so that reinforces why bark isn't usually desired in the composite. Then I assigned surface checks and voids in the samples before and after the sample was reconditioned. So we can compare the area occupied by the checks and voids before and after swelling, and also the irreversible thickness swelling. In this case, 19% for this sample. As you can see, numerous microchecks developed in OSB exposed to wetting and drying, as I hypothesized. So the area occupied by checks directly impacts change in dimensions. This graph shows the average checking and voids from 6 replicate samples from different panels. Checking increased significantly threefold, and voids increased significantly twofold. The average irreversible thickness swelling for the 6 replicates analyzed with this method was around 16%. Now I'd like to show you the internal microstructure of OSB probed by Helical X-ray CT. So after we load the datasets in the volume rendering software in the computer, we can then create animations, and with that we can segment, phase, and show them differently. In this case we are looking at the inside of the panel. We can also look at zinc in this case. We can also eliminate certain phases, like we're eliminating zinc here to enhance clarity of visualization of different phases. Here we're only seeing zinc before and after swelling. So after we segmented all the phases, we can analyze them individually, which gives us detailed information on what's happening in the OSB after swelling. This technique is the most time-consuming between all of them, but is the one that gives us the most precise information about the microstructure of the entire sample. So let's go to high magnification to have a look on the cracks and voids that were manually segmented. It takes around 2 weeks to assign one slice, but it's certainly worth it. So after we measure all of them, with image analysis we can gather data and run a statistical analysis. So from this graph we can see that out of the total sample now, the voids and the checks are a much greater proportion of the overall volume. Checking and voids increased significantly, whereas the strands and zinc decreased significantly. The only phase that did not change significantly was the cell lumen phase. And if we look at how that affects changing dimensions, out of 15% irreversible swelling, 12% is taken by internal checking. So finally, where does the fracture is occurring? With the field emission FE-SEM images, we sought to find that out. So as you can see from this cross-section of softwood, it's mainly occurring as radial delamination by interwall fractures at the interface between the wood cells called middle lamella, which is lignin-rich, and also in the form of transwall fractures. Here we can see a long check in the radial surface of softwood. And in terms of loss of zinc borate, note that the wet-dry dataset on the right suggests that wetting and drying reduced the level of zinc in the OSB specimen. However, a 3D network of fine particles of zinc borate remained. These results are in agreement with those from OSB exposed to water leaching published in this peer-reviewed paper which I co-authored. And also presented at the CWPA last year here in Vancouver. Now, I would like to summarize the main findings of this study. What is it happening when OSB swells? Do checks form? Yes, we did see significant checking of OSB. Unexpectedly, we found that checking was occurring during wetting when the OSB was swelling. That is the opposite for solid wood, which according to the literature only checks during shrinkage. Checking occurred less commonly in aspen flakes than pine flakes, as you can see on the flakes highlighted red, representing aspen. From this image, we can also see the partial recovery of deformed aspen vessels. Checking occurred on the surface and also deeper in the OSB, as you can see from the CT animation from the cross-section of OSB, before and after swelling at the same location from the datasets. So we can see that the checks go through the OSB and they get bigger after swelling. So as we saw previously, it's occurring mainly in the middle lamella. So now I would like to finish up this presentation pointing out some general conclusions and implications from this study. For the first time, significant microchecking of flakes in OSB was visualized. So from one of the first slides where I mentioned the factors in the literature that affect swelling of OSB, now it's possible to suggest that checking can also cause change in dimensions, such as in thickness swelling and linear expansion. Therefore, selecting woods that are less susceptible to checking, like I showed aspen versus pine, it can reduce swelling. And treatments that reduce checking of solid wood with water-repellent preservatives could possibly be used to reduce thickness swelling of OSB. With that, I would like to thank my supervisor, Dr. Phil Evans, and my committee members, Dr. Simon Ellis and Dr. Scott Reiniger. I also would like to thank the AWPA for granting me with 2 travel awards to travel down to the US and North Carolina and also Puerto Rico to present a portion of this work. And I also would like to thank my friends from my research group, which were vital supporting me during the course of this research, as well as people at the ANU who helped me collecting some data sets. With that, thank you very much for your attention.